All right, today we're going to dive into what is basically a genetic detective story. It's a fascinating case that starts with a family, a rare cancer, and a huge question that scientists were absolutely determined to answer.
So our story kicks off right here with this family tree. You see the filled in circles? Those are the people we're focused on. A mother, diagnosed at 34, and her daughter, who was diagnosed at just 7 years old. Both of them with the exact same cancer. The question just hangs in the air, right? Why them?
The diagnosis they got was Hodgkin lymphoma, which is a cancer of the lymphatic system. And while you do see it in younger people, having it hit a mother and a daughter, especially with the daughter being so young, well, that's really, really unusual.
So what's the deal here? Is this just a tragic coincidence? Some incredibly bad luck? Or is there something deeper going on? Something inherited? And this question is super important because it turns out the risk of Hodgkin lymphoma running in families is actually one of the highest for any cancer out there. This family could be holding a major clue. Okay, so this is where the real investigation kicks off. To find the answers, scientists went straight to the family's DNA. They basically treated their genetic code like a crime scene, and they were searching for that one single clue that could explain the whole thing. And the main tool they used for this? It's a technique called whole-genome sequencing. You can think of it like making a perfect copy of a person's entire genetic instruction manual. So by sequencing the mother, the daughter, and the healthy father, the researchers could put these manuals side by side and look for any typos. And listen, this is no easy task. The human genome has about 3 billion letters. Finding one meaningful mutation in all that, it's like trying to find one single critical typo in a massive library filled with thousands and thousands of books. The scale is just mind-boggling. So when you're faced with millions of potential clues, what's the first thing any good detective does? You've got to filter out the noise, right? The scientists set up this genetic funnel to narrow the search from millions of leads down to just a handful of prime suspects. And get this, they started with roughly 3.5 million genetic variations across just the 3 of them. That is an impossibly huge number of potential leads to chase down. So first, they tossed out all the common variations that you'd see in the general population. Then, and this was a really clever move, they got rid of any variants that the healthy father also had. And just like that, bam, the list of suspects plummets to just 1,331. For the final step, they focused only on the variants that were most likely to actually cause damage. You know, the ones that change how a protein works. After all that filtering, what were they left with? A very neat and tidy shortlist. Just 5 suspect genes. And here they are, the lineup. These are the 5 candidates that could be responsible. Each one is a solid lead, but the question is, which one is the real culprit? You know, every great detective story has that aha moment, that one big breakthrough. And in this case, it came from a source you might not expect, the grandmother. Her DNA was about to crack this case wide open. Remember that family tree we saw at the beginning? Well, the grandmother, at 66 years old, was totally healthy. She has the family's genetic history, of course, but she doesn't have the disease. That makes her DNA the perfect control, you know? It's like the genetic blueprint for a long and healthy life. And this is where it all comes together. It's brilliant. The scientists took those 5 suspect genes and just checked them against the grandma's DNA. 4 of them, they were there, present in her healthy genome, which probably means they're harmless. But one variant, one was absent. DICER1. It was in the mother and the daughter, but not in the healthy father or the healthy grandmother. The researchers put it pretty clearly in their report, basically concluding that this specific DICER1 variant was the most likely reason for the cancer in this family. They had their prime suspect. So, they've got their suspects. The next big question is, how did it do it? What does this gene even do? And how could one tiny mutation cause so much trouble? This image here shows us the gene itself, and that red arrow, that's pointing to the exact spot where this family's mutation is. And location is everything here, because this isn't just any old spot. It's in a highly conserved part of the gene that's basically its engine room. Now, to really get what DICER1 does, we gotta talk about something called microRNAs. The easiest way to think of these tiny molecules is like the brakes on your car, but for your cells. Their whole job is to tell cells when to stop growing and dividing to keep them from going rogue.
And here's the crucial link. The DICER1 gene is the factory that makes these microRNA breaks. So if you have a serious mutation in a key part of that factory, well, the breaks might not get made properly. And cells with faulty breaks? That is a recipe for disaster. But the case isn't totally closed yet. The detectives had a suspect and a motive. But now they needed a smoking gun. Direct, undeniable proof that this faulty gene was actually causing harm in the mother and daughter. And boom, here it is. This chart is the smoking gun. It is the final piece of the puzzle. It's measuring the levels of 4 of those important tumor-suppressing microRNAs, those breaks we were just talking about. Just look at the blue and green bars. That's the healthy father and grandmother. Their levels are normal. Now look at the orange and gray bars. That's the mother and daughter with the mutation. Their levels are way down. The breaks are failing. It's right there in the data. And this isn't just some random one-off finding. This discovery officially links this family's Hodgkin lymphoma to a known condition called DICER1 syndrome, which is a disorder that puts people at risk for a bunch of different tumors. This gives the family a definitive diagnosis and opens up a path for genetic counseling and future care. So let's wrap this case up. A single, new mutation in the DICER1 gene basically sabotaged the production of a cell's breaks. Without those breaks, cells were free to grow completely out of control, which ultimately led to Hodgkin lymphoma in 2 generations of the same family. The genetic mystery is solved. This whole story of scientific deduction is just incredible, and it leaves us with one final thought. This family found their answer buried deep inside a code 3 billion letters long. It just makes you wonder, doesn't it? How many other diseases, how many other family mysteries are just one single genetic clue away from being solved?